Hi everyone, today we are going to discuss about oxidative phosphorylation. Oxidative phosphorylation is the combination of oxidation and phosphorylation. The oxidative phosphorylation was explained by Peter Mitchell in 1961 and awarded the Nobel Prize in 1978. The oxidative phosphorylation is the process by which the energy stored in NADH and FADH is used to produce ATP. The oxidation step contains electron transport chain and the phosphorylation is the production of mitochondria have been termed as the powerhouse of cells since the final energy release takes place in the mitochondria only. Mitochondria have an outer membrane that is permeable to most metabolites and inner membrane that is selectively permeable, enclosing a matrix with it. The oxidative step consisting of electron transport chain and it is a series of protein complexes embedded in the mitochondrial membrane. And this chain consisting of 4 complexes and ATP synthesis. The 4 complexes are NADH, FADH, cytochrome B/C, cytochrome oxidase. In this step, electrons are come from this electron carrier and they travel through the electron transport chain where the electrons' final destination is oxygen, which will help to reduce oxygen to form water. So the oxygen is known as this final acceptor. Electrons captured from these donor molecules are transferred through the 4 complex. And the chemosmotic theory was developed by Peter Mitchell, which explained the mechanism of ATP formation. According to this theory, the transferred electrons down an electron transport system through a series of oxidation reduction reaction release energy as electrons are transferred along the electron transport chain from electron donor to electron acceptor in the inner mitochondrial membrane. Free energy is released. This energy allows certain carriers in this chain to transport hydrogen ions, protons, which thus contains a higher concentrations of protons than the matrix. This creates an electrochemical gradient across this inner membrane. The energized states of these membranes are as a result of this charge separation is called proton motive force. And this proton motive force provides the energy necessary for the enzyme called ATP synthesis and to catalyze the synthesizing of ATP from ADP and phosphate. The generation of ATP occurs as the protein across the membranes through the ATP synthesis complex re-enter the matrix of this mitochondria. As the protons moves down the concentration gradients through the ATP synthesis, the energy released causes the rotor and stalk of this ATP synthesis to rotate, and the mechanical energy from this rotation is converted into the chemical energy as phosphate is added to the ADP to form ATP in the catalytic head F1 domain. Phosphorylation step. ATP is generated by the phosphorylation of ADP. The ATP synthesis has two subunits, F1 subunit and FO subunit. Several subunits of this proteins forms a ball-like shaped arrangements around the axis known as F1 and which project into the matrix and contains the phosphorylation mechanism. The transmembrane FO subunit which contain a protein channel for the flow of protons. The hydrogen concentration is much greater in this inner membrane space than in this matrix, thus the generating an electrochemical proton gradient, and this gradient derives the protons back across in the inner membrane through the ATP synthesis that catalyzes the synthesizing of ATP from this ADP and the inorganic phosphate. The flow of protons through FO causes it to rotate, deriving the production of ATP in the F1 complex.